Protandim contains 5 phytochemicals, and again, the structures are not important to memorize, so you'll be relieved at that. But they come from 5 different plants. All of these plants have a long, long history in traditional medicine. People didn't know how they worked, but for centuries they knew they do work. What they didn't know was that they are regulators of a protein called NRF2 and activators of NRF2. And that's the mechanism whereby more than 500 of your most protective genes are regulated by these simple, believe it or not, chemicals produced by humble little plants that have been recognized in India and China for centuries and centuries. They activate what's called the master regulator of stress-related responses, of antioxidant enzymes. How does Protandim activate NRF2? And you hear a lot about NRF2. You're going to hear even more about NRF2 as time goes on. How does Protandim activate NRF2 in a cell? This is a cartoon that illustrates that. This purple shape in the upper right corner of this slide represents one of those 5 chemicals from plants, from medicinal plants. And so as Protandim approaches a cell, the cell actually has receptors on its surface. And my analogy here is this is like if the cell is your house, Protandim just stepped onto your front porch. And it rings your doorbell when that happens. And that causes a change inside your house. You hear the chimes or the bell. And you know something has just happened that may change events within your house. In this case, an enzyme on the inside of that cellular house called a kinase has been activated, signified by turning yellow. Well, the business of that kinase is to put phosphate groups, to chemically modify a handful of molecules that it knows how to modify. One of those is that little red oval labeled NRF2. NRF2 is being held in the cytosol, outside the nucleus where the DNA resides, by this other larger protein called KEAP1. So the activated kinase now chemically modifies NRF2, and you can see that little yellow P that's been added. That's a phosphate group. So it's chemically changed NRF2. And the protein that's holding on to NRF2 now sees this thing has changed its shape. It doesn't fit my grasp quite as well. It releases that NRF2. So it's now free to diffuse around the cell, and it's small enough it can actually enter the nucleus, which is the vault. It's where all the plans, the blueprints for everything you are reside in the form of DNA. So this activated NRF2 now has access to all of those blueprints, and it binds to specific genes within there. Out of 25,000, at least 500 or 600 bind activated NRF2, and the result is those genes are turned on. Each of those genes has a switch, like a dimmer switch on the lights in your house. NRF2 simply turns that knob. Those genes express what they encode. The products designated by the blueprint of each and every of those genes is now produced. And among those products are antioxidant enzymes, anti-inflammatory enzymes, antifibrotic enzymes, all kinds of stress-related genes that help cells, especially when they're in chemical stress, when they're in oxidative stress. It helps bring things back into balance and to provide a normalizing influence on those cells. We can visualize this microscopically. The upper 3 panels show control cells, cells without Protandim. And here, again, I won't go into a lot of detail, but we can stain NRF2, and it stains red in this slide. It's a fluorescent red stain for NRF2. We can also stain the nuclei of those cells. It's staining the DNA. And if you merge those two, you get what you see in the upper right. And it may be a little hard to see in this lighting, but what you see if you look carefully at that is the nuclei are green and the NRF2 is red surrounding the nuclei. It's not where the DNA is. If we treat those same cells with Protandim and now stain the NRF2 and stain the DNA in the nucleus and now superimpose the red stain and the green stain, what you see is the lower right figure. Everything looks yellow, and that's because if you add red light and green light together, what your eye sees is yellow. This proves to scientists looking through this microscope that Protandim treatment caused the nerve to go from its location outside the nucleus in the upper row to exactly where the DNA is. That is, the NRF2 has moved into the nucleus, and that's where it can regulate those genes, turn up all of those processes. The first paper published in 2006, I've condensed into one or two sentences for you here. This is really what you don't need to know about everything in that paper, but you do need to know this. After 30 days of supplementation, T-bars, which is oxidized lipids, declined by an average of 40%, and the age-dependent increase was eliminated. By 120 days, SOD and catalase had increased by 30%, 54% in the red cells, respectively. That's very significant. You have all seen this figure. This shows what T-bars looks like in a population of varying age. There's a lot of scatter. We're not all the same at age 40 or 50 or 60. Some of us are in great shape. Some of us are already suffering from diseases associated with aging. If you draw a linear regression line, the line through those scattered dots, you can see that there's a relentless progression as you go from 20 to 80. Your T-bars averages go up and up and up. If you look at people in this group who were taking conventional so-called antioxidant supplements that many of us took for decades, those are the points that turn from red to blue. So some of these people were taking about 0.5 gram of vitamin C every day, about 400 units of vitamin E. And you might think they would have been better off than their colleagues who weren't taking those, but they weren't. Those blue dots, in fact, if you draw a linear regression line, fall on a line with an even steeper slope. And you've heard a lot in Time and Newsweek and on the evening news, studies that show traditional antioxidants, antioxidant vitamins don't always help. Sometimes they can even make things worse. Well, indeed, that's true.